Well, thanks, biocrates, for having me. It's kind of unusual for me as a clinician to speak in a really basic science audience, but I will do my best to to answer all your questions. So, as I already said, I'm a consultant for hepatology in the Medical University of Innsbruck. And my main focus in science is on host microbiome interactions, gut interactions, and I focus there mainly on the liver. So why is that? I can't move on.
Okay. Maybe if you click, sometimes it reactivates things.
No. Okay. Well, the liver-gut axis is the main focus of my scientific research, and it's really logical if you want to look at it that way, because the liver is the organ system who is next to the intestinal gut, and all the metabolites, all the nutritional factors, and so on are going via portal vein into the liver. And so we have the picture of bio multilateral interactions. There are signals from the gut who affect the liver pathway, e.g., for example, microbiome or nutrient-derived, as I already mentioned. But on the other hand, there are also liver-derived signals affect gut processes, like bile acids or immune mechanisms. Prototypic example for microbial intestine-driven pathway is like nutrient-affected pathways. It really depends how your microbiome looks like on what you are eating and what your diet is, and it's also affecting the liver, which I would like to explain later on one of the main diseases causing worldwide called metabolic associated dysfunction. Liver disease already causes affecting 40% of all patients worldwide. So this is a kind of, can I go back? Sorry. This is a slide which is kind of complicated, but it should illustrate what is really important for me. It's like you have already mentioned, or Alice has already mentioned, that all organs interact with each other. And so we are talking about the microbiota, and we are talking hereby about the gut microbiota. Also skin or the dermatological side has a real influential microbiota, but my focus lies on the gut microbiota. And it lies in the composition of the gut microbiota, how, as you can see here, how the gut barrier works or doesn't work. So if you have a leaky gut or if you don't have it. And it works by receptors like toll-like receptor 4 or GPR41, and it sends mostly metabolomes like VLDL and gut nutrient-derived signals like free fatty acids or fructose into the liver and into the adipose tissue, changing cytokines and chemokines and therefore interacting with the liver. As you can see, this paper is not new, it's from 2010, but on the understanding of how these three organs interact, nothing has changed really. So if we look closer to microbiota, we have about 100 trillion bacteria in our gut. It's a biomass of 0.5 kilo or 1 pound. And as I already mentioned, it affects crucially the local and systemic immunity. Critically involved in metabolic processes. So if you have changes in your gut microbiota, mostly affected by westernization of the diet, you have metabolic diseases like fatty liver disease, which is my main topic, or cardiovascular disease, which may be the topic of our last speaker, and stroke and things like that. And it plays a really major role exactly in GI and liver disorders. And the microbiota is affected by many co-founding factors, but I would also mention again the dietary factor because it's the most important. So if we look into our intestinal system again, we are a composite of species, a superorganism. The microbial census exceeds total number of human cells by 10-fold. So it means we have 10 to 14 bugs. The largest collection of microbes resides in the GI tract. And we called it's really important to understand the difference between microbiota, microbiome, and human metagenome because it leads to the host microbial interactions. Microbiota and all organisms living in a circumscribed area. The microbiome, the aggregate genomes these species encode. And the microbiome is an integral part of our genetic landscape and is called our human metagenome. So how many if we talk about microbiota, I always have to mention we are talking here about prokaryotic microbiota. Fungicides and viral microbiota is not included. So the main part of all our microbiota is located in the colon with 10-fold 14. And if you see it on this slide, from the oral cavity to the anus, there you have more and more microbiomes living inside your GI tract. So here you have 10-fold 11, and here you go by 10-fold 14 microbiomes living in your GI tract. So what is the structure of the intestinal microbiota? That's really important. You have, if you look at the GI tract, you have a loosely adherent mucosal layer about 450 micrometers filled with microbial prokaryotic cells. And then you have a layer with firmly adherent prokaryotic cells. Then you have the glycocalyx, which just sticks really close. And then you have the epithelial and the dendritic cells and the lymphocytes who are transporting or helping transporting things also via here the gut blood barrier into the blood. And there, these cells, the lymphocytes and the dendritic cells, are of course for immune modulation the most important thing. So what is really important is the composition of the intestinal microbiota is relatively stable. The difference is mainly at the levels of species and families. And it's a really complex analysis, as you all mentioned and we all know, because there are so many different microbial prokaryotic cells in our GI tract. So if you go down, where do we start? We start by the life and the domain and then come to the kingdom. I have chosen two really pathogen bacteria, Clostridium difficile, causing severe diarrhea, and methicillin-resistant Staphylococcus aureus, causing all kinds of infection, mostly not in the GI tract, mostly in lungs and stuff like that. So they are both bacteria, of course. The phylum is also the same. They are both Firmicutes. And the class, here it starts to differ. There you have Clostridia and here you have Bacilli. And the order going down to Bacillus and Clostridiales, and then family. So this is the main part where do you have changes. These parts up there are really stable across diseases in the microbiome. The family, genus, and species, they differ. So you more or less have different cases or different numbers of Staphylococcus and Clostridiaceae, of Staphylococcus and Clostridium, and other causes of Staphylococcus aureus and Clostridium difficile, where you are in the species level. The problem with microbial research is that you are always most interested in the species because they are the easiest to attack kind of or change or in a way where you can do it in a clinical setting. It's really hard to interact with class or order. And this is one of the main problems of microbial or clinical microbial research. So we have a big change in taxonomic diversity, where there are over 5,000 species, and what you see, there are a lot of them are Firmicutes and Bacteroidetes. If you have these two kinds of phyla, you're already covering about 90%. Everything else is more or less not that common. What is really important that every human being has like a fingerprint, a really individual microbiota, and this microbial stability is really astonishing because there are only really a few changes possible, and most of these effects that changes are possible are on a dietary pathway. Everything else is not changeable. So why is that so important for us, for clinicians, because if you have obligate anaerobic bacteria like you have here, you can see, as I have shown you previously with all the layers, you have floating them more or less here around, and then you have the interaction between the intraluminal bacteria and the host here with the blood vessels, and everything is fine. And here metabolomics are really important, like butyrate and nitrate and things like that, that the T cell regulation and the immunity is kind of stable. But if you have pathogenic bacteria, and they are mostly not pathogenic bacteria like you know them, like Clostridium difficile, they are mostly, as I already mentioned, triggered by nutrition. And then this kind of balance between the microbiota and the host, it's not there any longer. And then you see the butyrate is going down, nitrate is going up, SCFAs are going down. And if it is like that, then you become something that we call leaky gut or impaired gut barrier. And if you have an impaired gut barrier, pathogenic bacteria will go into the host and therefore activate inflammatory pathways. And this is not only important for the liver, because that's important for me, it's also important for the brain, which will be the talk about our next talk. And then inflammation in muscle, it doesn't matter if it's the heart muscle or other muscle, and adipose tissue. So I will go a little bit deeper into the pathological area of the microbiota. And for me, my main research is metabolic dysfunction associated steatotic liver disease. First, I just want to mention why is it so important, because as I've already said, 40% of all people around the world are affected by MASLD. And it's not only inflammation of the liver, it interacts via metabolomics and microbiota with all other systems, like the cardiovascular system for one of the main points, the adipose tissue, and the cholesterol levels and stuff like that. So it belongs to the metabolic diseases and therefore has a really high incidence and prevalence. But to go back to the basics, there you see we had done with a Chinese group together an experiment on a murine level, and we had OB mice. And if you give OB mice probiotics, that means you change the microbial composition. The ALT, one of the main markers of MASLD disease, goes down. It decreases. And you also see here in the histological aspects that the fat, which is here white, goes down in the liver as well. And this was a groundbreaking paper, and Lombard et al., Lombard is one of the main players in MASLD and NAFLD, as we have called it till 2023, scientists. And it is really important that NAFLD or MASLD is at the moment, or has been only be possible to be diagnosed via biopsy. And that's kind of a weird thing for a clinician, because you have to take a histological sample from inside someone's body, and it's not what we want to do because we can harm people by that. So our goal is always to have some non-invasive tests. And Lombard et al. Found that the gut microbiome-derived signature with high levels of proteobacteria and E. coli and low levels of Firmicutes is specifically for NAFLD. And he could do that to show that it's possible to detect these microbial signatures in the stool of patients, and therefore can detect NAFLD without invasive tests. What is important? As you see, we have like some bacteria who are more abundant, like proteobacteria, and there is a decrease in a lot of them, like Fecalibacterium prausnitzii or Bacterium recale and Rhinococcus obeum. Why is that so important? Because these bacteria, like Fecalibacterium prausnitzii, has anti-inflammatory effects, which have been established in many other murine models. And so it shows that all the changes in the microbiome are infecting inflammation, systemic inflammation in the host. This led to another study, and this study had a really big population for clinical studies. It had 1,282 people, and these people were followed up for 5 years. And not only that, there was an extensive characterization done of these patients with interviews and anthropomatics and lab tests and pancreatic elastase and liver and carotid ultrasound. And all these patients received RNA gene sequencing of fecal samples at the baseline of the study and 5 years afterwards. And what could you see? That patients with an unstable intestinal microbiome, that means they have a lot of changes between the 5 years, had really high, they were really likely to become fatty liver diseases, hypertension, and diabetes mellitus, which are all diseases associated with the metabolic disease group. So if you look here at this figure, you can say that Clostridium difficile or Clostridium bacteria, who are already known as pathogens, are really high in these patients who establish disease. And if you look on a metabolomic pathway, you can also see that here lysosomals, which is mostly associated with free fatty acids, as you may all know, are going up. And so we can see here in this study, in a big cohort, in a human cohort, that the metabolome, the microbiome, are acting with each other. And this is another study which is also highly published, showing that it is a difference if you have patients with fatty liver with morbidly obese patients, which is the main part of these patients, and not morbidly obese patients, showing here that you have a significant different microbial analysis. And you also see here, if you look at these analyses, that the Shannon index and the PCOA, the biocrates index, is completely different from people who are severely obese and patients who are not severely obese. This goes along with what I've talked earlier about, that the adipose tissue, the liver, the microbiome, the brain, and the heart, they are all interacting. So these gut-derived metabolites, that's what I'm talking all along, are the drivers of disease. And you have these gut microbiota-associated factors, and there are a lot of them, like short-chain fatty acids and LPS and lactate and ethanol and FXR. And all these factors lead to a progression of NAFLD from a normal liver to MASLD and on, so a cirrhotic liver. As I've already mentioned, it is possible to use these findings for a therapeutic intervention in muscle therapy, and therefore you would like to change the gut microbiome. Mainly you do it at the moment with personalized nutrition, but in the future there would be other options, maybe like FMT, which means stool transplantation, or the bacteriophage therapy, which I think would be the most elegant, but it's only at the basic science level at the moment. And by changing the gut microbiome, you change the microbial metabolites, like I've already said, SCFAs and BCAAs and indoles and secondary bile acids, and also microbial products like LPS, and therefore you do not activate TLR4, and therefore you don't create inflammation in the liver. So here is another slide showing the same thing I've talked all along, the high interaction between microbiome and the host. They are here in the liver, and that you understand here on adipose tissue. You see increased FEAAs. There is an influence between the liver and the muscle and the adipose tissue. it also goes along with amino acids and high glucose and triglycerides, and it all leads in the end, or it starts, we don't know that yet, with insulin resistance. One of the last papers published that are really highlighting in this field of research is that we have kind of a clinical score called FIB4, and a lot of people are using them because it's a really good marker for fatty liver disease. And this paper showed that metabolomics, especially lipidomics, are more precise than this FIB4 score. And this FIB4 score had a prevalence and incidence about, I think it's about 90%, so it's really good. But lipidomics are much better. And so I think it is a way how future will be created and how the future will look like by defining metabolomics who are specific for different diseases. As I've already mentioned, nutrients are the major player in this axis, and I would like to show you one study. It's a real cool one. They only have included 10 patients, and they did a keto diet, as we all know. So all these patients were not allowed to eat carbs. And then there was a broad analysis, including metagenomics and metabolomics and transcriptomics. And as you can see, this low-carb diet improves liver fat metabolism. It promotes rapid shifts in the gut microbiota composition. It induces microbial changes with increased circulating folate. And this also leads to folate-dependent 1-carbon metabolism gene expression in liver, meaning that the liver is trying to heal. So if you look at the benefits of the carbohydrate-restricted diet on a microbial level, you see as here, here you have the more pathogenic microbial composition, and in 14 days you can switch to a really anti-inflammatory microbial composition. And I would like to show you one more study, and then I think I'm finished. You can see it depends on what you are eating, and then you can produce, especially in patients with diabetes, you find a lot of histidine producing imidazole propionate, and this impaired glucose metabolism. And as you can see here, imidazole propionate is increased in individuals with diabetes 2. And this is important because imidazole propionate is one particular sign for metabolic diseases, and it's produced from histidine by type 2 diabetes associated bacteria, and it impairs glucose tolerance and insulin signaling, and it inhibits all this activation via mTORC receptor. And another study shows that the gut pathobiont Enterococcus gallinarum triggers autoimmune response dependent on a genetic background. And I think this is really, really, really important because it shows that our microbiota not only interacts with our organic system, it also acts with our autoimmune system, and it also interacts with our genetic background. And so I think the metabolomic genetic pathway is really important. And last but not least, I want to talk really, really shortly about the bile acids, because they are kind of liver produced and interact with the microbiome as well, the other way around. So not the microbiome is influencing the liver is influencing microbiome. And you see that depending on glucose metabolism and changes in your microbiota, there you have changes in the bile acids. And bile acids activate FXR. And FXR is really important for inflammation signaling, especially in the liver. And FXR stimulates, on the other hand, in the terminal ileum FGF19. And FGF19, I think, is one of the major fibroblast growth factor is one of the major players in metabolic diseases. So you can see by changing the liver via the metabolic pathway, they influence the microbial composition and interacts with the gut, and therefore increases inflammation in the human body. And there is one study that shows how you can interact with these systems, and this is a medication that is kind of proven already in primary biliary cholangitis, and it shows that if you take it in steatohepatitis and therefore reduce bile acids and the metabolomic effect of bile acids, you can see how you have clinical changes in lab chemistry according to the liver, and you also can see that you have a going down of the fatty liver in imaging. Last but not least, we have non-microbiome-derived intestine-derived signals in the gut-liver axis, as I've already mentioned, FGF19. And here you can see that these metabolic effects of FGF21, 15, and 19 interacts with all our systems, not only with the liver. And these things have already been in clinical studies as well, where you have these metabolomic products like FGF, and if you can stop them or suppress them, you can show that the fatty liver or the lab parameters of fatty liver decrease significantly. And so I come to my conclusions. Gut-liver axis is the prototype of microbiome-host interaction. Gut-liver axis plays a crucial role in liver diseases and all other diseases. And gut-liver axis is a valuable clinical target in the future. Thank you.